Okay, welcome back. In this video, we will cover Chapter 3, Biochemistry, the Basic Ingredients of Life. Now, chemistry is the study of atoms and molecules and how they interact with one another. And in physiology, it's largely about the interactions between the molecules in our cells and in our tissues, and more specifically how chemistry relates to organisms. At the root of all metabolic reactions is chemistry. So understanding how chemistry works will help you understand more clearly how the body works. And there's a special division of chemistry called biochemistry. Bio meaning life. So when you're talking about the chemistry of a living organism, such as a human, you're talking about biochemistry. To fully understand how a body works, or to understand what we'll talk about in future chapters in this course, you need to have at least a basic understanding of some fundamental biochemistry. Okay, learning objectives for this chapter: to be able to differentiate between atoms, elements, and ions, to define pH, acids, and bases, and what their roles are in the body, describe chemical bonding and the role of water, be able to describe the properties of a solution, and being able to distinguish among the different types of biological molecules, explain metabolism, explain cellular respiration, and finally to explain the role of enzymes in physiology. Alright, now we'll talk about atoms, elements, and ions. All matter, no matter if it's living or non-living, no matter how simple, no matter how complex, is made up of elements. And an element is the smallest unit that retains the unique chemical properties of a very particular type of matter. And also, very important for elements, they cannot be broken down into anything more simple. So if you have a mass of, say, calcium, if you were to break that up, you would just get smaller parts of calcium. You don't get calcium and something different. You get smaller and smaller amounts of that basic fundamental type of matter, the element. It can't get any simpler than an element. Okay, elements are usually abbreviated by the first few letters of their technical name. And I say usually because this is not always the case. For some elements it is pretty straightforward. C for carbon, O for oxygen, H for hydrogen, and so on. And there are certain rules on how you can name elements. They're going to be either one or two letters. And if you use two letters, the first one is always capitalized. But for some elements, the chemical abbreviation is the elemental abbreviation is from its Latin or its Greek name. So the symbols don't seem to match up with the element. And a good example is sodium. The Latin name for sodium is natrium. The symbol for sodium isn't just S or SO, it is Na for natrium. These are just some things that you just have to learn. Something as simple as chlorine would be Cl. Potassium is another good example. Potassium's symbol isn't P, it is K for its Latin name. Whenever you have two or more elements joined together, they form a molecule. And some examples of molecules: a compound is when you have more than one type of element. A good example, or a very common example, is water, H2O. You have hydrogen and oxygen, two different elements combining together to form a compound. And one example we'll talk about in this chapter and also in future chapters, sodium chloride, NaCl, which is the common example of table salt, the sodium chloride. Alright, now we'll talk about the atom. It's the smallest recognizable unit of any element. And atoms consist of a nucleus, and within that nucleus, the nucleus is considered the command center of that atom, the brains of the atom. Within that nucleus you have different types of particles, and they have different types of charges. In the nucleus you find a proton, which has a positive charge, and you have neutrons, which have a neutral charge or no charge. So P for proton, P for positive, N for neutron, or N for neutral also. And the particles that orbit this nucleus are the electrons, and they have a negative charge. So the electrons basically orbit the nucleus like the planets orbit the sun. Right, here are some common elements that are required in your daily diet. Iron, symbol Fe, because of its Latin name ferrous, and how much your daily requirement should be, and its role within the body. And also what happens if you get too little of each individual element. So you have iron here, you have copper, abbreviated by symbol Cu for copper. Co would be cobalt, but Cu is copper. Zinc, manganese, iodine, and then fluorine. All very common elements that are needed within your diet every day. Alright, here are some illustrations of hydrogen, helium, and lithium atoms. We'll go to lithium here. Its atomic number is 3. It's going to represent 3 protons and 3 electrons here, and there are 4 neutrons inside the nucleus. This whole area right here will be the nucleus. The pluses indicate the positively charged protons, and these yellow circles that have no marking on them at all represents a neutral charge, or the neutrons. And then these 3 negatively charged particles which are orbiting the nucleus are the electrons. So in this illustration, lithium has 3 electrons here, helium has 2, 1, 2, and hydrogen has 1. Now in a typical atom, the number of protons, which are positively charged, will equal the number of electrons, which are negatively charged. So they balance out. So in a typical atom, there's going to be no charge. It's going to be a neutral charge. But there are many cases where electrons are either gained or either lost. And by doing that, you are making this balance unequal. So sometimes atoms will have a positive charge, sometimes atoms will have a negative charge. In either case, they're referred to as an ion. An ion means any atom that has any charge to it other than neutral. It could be a positive charge or it could be a negative charge. But in either case, that is an ion. Now something that seems counterintuitive at first, when an atom loses an electron, it becomes more positively charged. And the opposite of that, when an atom gains electrons, they become more negatively charged. And at first that seems backwards. You would think losing something is bad and gaining something is good. But think of what's being lost and think of what's being gained. You're talking about a particle with a negative charge. So if you have more of those, if you gain more of those, you're becoming more and more negative. If you lose those negative charges, you're becoming more positive. So it's very easy to get this relationship backwards. If you gain electrons, you become more negative. If you lose electrons, you become more positive. And a good example here is sodium chloride, table salt that we talked about a minute ago. The compound itself, sodium chloride, has a neutral charge, but the individual components are not. If sodium chloride were to break up, or to be separated, what's called ionization, you're forming 2 separate ions. The sodium ion, which is positively charged, as I see the plus here, and chloride, which is a negatively charged ion. That's an ion, that's an ion. But when they are joined together, that negative charge and the positive charge balance each other out, so the compound of table salt has a neutral charge to it. Right, ions are also considered to be polar because they have charges on them, like the poles on a battery or on a bar magnet. The positively charged atom will be attracted to the negatively charged atom. So think of using a bar magnet. If you've ever played with bar magnets before, if you were to put a one pole versus another pole side by side, if they are opposite charges, they will attract each other. If they are the same charge, they will repel one another. You can actually feel the repulsive force between them. You can almost move one magnet with the other because they can't attract one another. So opposite signs will attract one another, and conversely, similar signs will repel one another. Right, another type of ion is called an electrolyte. These are charged ions that are found within the body, and they are very influential on muscle activity, the nervous system in general, and fluid balance. And they're called electrolytes because they're able to conduct electricity. So you can imagine that's going to be very important for the nervous system and very important for muscle activity also. Here's a list of some important electrolytes. Sodium, Na+, potassium, symbol is K, calcium, Ca2+, magnesium, chloride, hydrogen phosphate, bicarbonate. These are all electrolytes. These are all ions, positive or negative. These are all ions that are able to conduct electricity. Here's a standard picture of the periodic table of elements. And a lot of times you'll see it color coordinated based on the group or the properties of that grouping of elements. And also these 2 rows that are here, they normally would fit in here, but in order to make the table more legible and prevent it from having it be way too big, these 2 rows are removed out from here to here. That's why the numbers jump from 56, 57 up to 72. Those ones that are missing are down here. Same thing down here. From 89 jumps to 104, but that row is inserted down here. And actually, fairly recently, these last 3 elements have been named. So there's no unnamed element on the periodic table as of 2016. All right, next we'll talk about acids and bases. These are also considered electrolytes because they are able to conduct electricity. And they're also able to break down or disassociate in water. Now, acids will dissolve metals and can actually burn your skin and also burn holes through metals, depending on how strong they are. The definition of an acid: this is a chemical that will release hydrogen ions. All acids have a set of very typical characteristics. They will taste sour. When they dissolve in water, they release hydrogen ions, and they are easy to react with other atoms. The bases, on the other hand, will have a very bitter taste, and they will accept hydrogen ions. Common example: bicarbonate, HCO3 with a negative charge, will easily accept a hydrogen ion, forming a carbonic acid, which is a very weak acid, and that's very important when it comes to controlling the rate and depth of your breathing, and that's something we'll talk about in more detail in a future video. Hydroxides, OH-, are very common bases, which are also able to accept hydrogen ions. So a good way to keep these straight: acids will release hydrogen ions, H+, and bases, when they break up, will release hydroxide ions, or OH-. Alright, the concentration of acids and bases are measured on what's called a pH scale. And this is a number scale from 0 to 14. And these numbers will represent the amount of hydrogen or hydroxide within that compound. The lower numbers, 0 to 6.9, are considered acidic, because they have more hydrogen ions. And the lower the number you get, the stronger the acid is. So an acid with a pH of 1, such as hydrochloric acid that's found in your stomach, is very, very strong. It can burn your skin if it touches your skin. And that's why heartburn actually hurts so bad, because that's hydrochloric acid burning through tissue. A pH of 7 is considered to be neutral. There's the same amount of hydrogen ions versus hydroxide ions. And a good example of this is distilled water. It's not acidic, it's not basic, it's just neutral. And the numbers that are higher than 7, 7.1 up to 14, are considered to be basic or alkaline or alkalotic. Either term is acceptable. And in this case, you have more hydroxide ions than you do hydrogen ions. There are many systems that are found within the body to help to regulate acid and base balance. The amounts of acids versus bases in the body needs to be regulated and controlled pretty tightly. Otherwise, you'll get very, very sick very quickly. See, the respiratory system takes in air and gets rid of carbon dioxide. That process we'll talk in further detail later, or why it's important. Because the carbon dioxide is a very weak acid. If you stop breathing, say if you hold your breath, for example, carbon dioxide will build up in the body, and your body can't take too much of that. When there's a buildup of this acid detected within the brain, there's an overwhelming desire to breathe. So whenever a child throws a tantrum and threatens to hold their breath until they get what they want, you might as well just let them, because their body won't let them hold their breath forever. They will eventually get to the point where they will either pass out and start breathing again on their own, or their body will be forced to breathe for them. Okay, the acid-base balance is also critical when we talk about the kidneys, and how they're able to deal with the excretion of excess acids within the urine. Another topic we'll talk greater detail about in a future video. Two of the many systems that are used within the body to help monitor the concentrations of acids versus bases. Alright, here's a drawing of a pH scale. See, we start down here with the very low numbers. Remember, the lower the number, the stronger the acid. So hydrochloric acid, roughly between 0 to maybe 1, very, very strong acid. And as you get closer toward the neutral point, you get weaker and weaker. So hydrochloric acid would be a strong acid. Wine is a little bit weaker. Black coffee is a little bit weaker. Urine is a fairly weak acid. Then you have 7, which is neutral, such as cytoplasm within cells. Distilled water is neutral. Then as you get higher and higher, you're getting stronger as a base. Pancreatic juice with a pH of 8 is a weak base. Household bleach is a little bit stronger. Ammonia is a little bit stronger. And sodium hydroxide is a very strong base. So the extremes on either end represent the strongest ones of those. So pH of 0, very strong acid. pH of 14, very, very strong base. Our elements can be joined together to form molecules. And individual elements and molecules are held together by bonds between electrons in the atoms. And that's why some elements will be quick to lose electrons. Some elements are quick to gain electrons. It's all about how elements join together and form bonds, which will form molecules. Right, if there's one atom that will donate its electron, you are forming an ionic bond, because when they lose that electron, they are creating an ion. That's why this is called an ionic bond. And a good example is one we've talked about already, sodium chloride. When these two are separate, You have the sodium ion and chloride ion. These are both very reactive. Sodium is very very quick to donate its electron to make itself more stable, and chlorine is very eager to accept an electron to make it more stable. That's how these two will join together. Because one of the key things in chemistry is being stable. Elements and your body in general will do whatever it can to become much more stable. For some elements, that means losing electrons. For some elements, that means gaining electrons. So sodium wants to give up this one on its outermost shell. Chloride wants to gain that one to fill out its outermost shell. So when they join together, they are forming sodium ion and a chloride ion, and when they join together, form an ionic bond, which gives you sodium chloride or table salt. Alright, if electrons are shared by the two atoms, a covalent bond is created. Now sometimes this sharing can be unequal because one atom will take a little more of its share than it should. And this type of covalent bond is called a polar covalent bond because you are creating areas of that compound that are differently charged. Some areas are going to be weaker, some areas are going to be stronger. Now polar covalent bonds are going to have a slight charge to them, a little bit more polar than a regular covalent bond, but less polar than the higher charged ionic bonds. Here are some examples of some covalent bonding, both with the single bonds with hydrogen, H2. We have a double bond formed between oxygen atoms, O2, and in a compound of methane, you have one carbon and 4 hydrogens on all the way around it. All those are sharing electrons, and the sharing makes each individual component more stable and makes the compound in general more stable. Alright, here's a summary of ionic bonds, polar covalent bonds, and covalent bonds. And what happens to their electrons, if they're shared or not, is there a charge, and a common example. So ionic, electrons aren't shared. Basically, one gets donated or stolen by another one. And by doing so, you are forming ions, such as table salt. Polar covalent, you have an unequal sharing. One will hog electron a little bit more than the others. So there's going to be, you have some areas of the compound that are going to be more negatively charged. Some areas are going to be more positively charged. A good example of that is water. And covalent is going to be an equal sharing of all the electrons, so there's no charge created, and carbon dioxide is a good example. Alright, next we'll talk about water, H2O. As you can imagine, the chief liquid that's found in all biological systems. All the fluid in your body is water-based, whether it be the plasma in your blood, or cerebrospinal fluid, or lymph. It's all water-based. Water is going to be a polar solvent. Bonds between the hydrogen and oxygen within water are going to be polar covalent. And that's because you have oxygen within that compound of water will take more of its share of the electrons. So this will create a charge on the hydrogen end, which will be a little bit different than the charge on the oxygen end. This is also how water molecules will bind with other water molecules. The positive end of one will be attracted to the negative end of the other water molecule. Alright, charged molecules that contain elements like oxygen and phosphorus and nitrogen will mix very easily with water. These are called hydrophilic, or water-loving. Hydro means water, and philic means to love. There are other molecules that don't mix well with water, such as oils and fats. These are called hydrophobic. Hydro, water, and phobic meaning to fear. So oil and water chemically actually do not mix. They don't do well together. Here's some examples of carbon dioxide up here. One carbon and 2 oxygens. Here you have an example of water, H2O, and 2 H's, the oxygen. When you look at the compound in general, the hydrogen ends will be more positively charged. The oxygen end will be more negatively charged. That's why these bonds are considered to be polar covalent. The electrons are being shared, yes, but they're not being shared equally. So you're creating an imbalance of charges throughout the compound. Now that polarity of the water will cause development of hydrogen bonds between water molecules. Is where you have the hydrogen of one water molecule will bind very weakly to the oxygen of another water molecule. Okay, so these weak bonds between these water molecules will actually increase the water's heat capacity. That means the water is able to store heat. Water will heat up and also cool down more slowly than air. Alright, now I'll move on to solutions. This is when one substance is dissolved in another. That combination together is called a solution. The substance that's being dissolved is called a solute, and the substance doing the dissolving which in almost every case in the human body will be water. The water is called the solvent. So if you were to mix, say, salt with water, the salt would be the solute, water would be the solvent. Now, electrolytes are the solutes dissolved in your water-based body fluids. And we've already talked about some important electrolytes earlier. Potassium, calcium, phosphorus, so on. Now, the amount of solute dissolved in the solvent is called the solute concentration. How much of that stuff is there? Now, how much potassium is there? How much calcium is there in relation to the overall sample? All right, now we'll move on to biological molecules. Now, most of your anatomy is based up of biological molecules. And biological molecules are found in living systems will contain some very common elements: carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus, and so on. But those are all very commonly found elements within a living system. Now, all biological molecules within your body and in a living system in general will fall into one of four categories. It's either going to be a carbohydrate, a lipid, a protein, or a nucleic acid. First, we'll talk about the carbs, the carbohydrates. These are sugars and starches. A starch is really just a complex sugar. And whenever you hear the word carbohydrate, all sugars fall under carbohydrates. So you can interchange these two words here. And these are the molecules that will supply energy for the living organism. And if we were to break down the word carbohydrate, it translates into a watered carbon. Now, all carbohydrate molecules all have a very typical ratio between carbon, oxygen, and hydrogen in them. And it's going to be one carbon to two hydrogens to one oxygen. It's always going to be in that ratio. So for here, C1, even though the 1 isn't written, it's understood to be a 1. So C1H2O1. Going up from this, C6H12O6. The ratio is always going to be the same for all carbohydrate molecules. Now, the very simplest sugars, monosaccharides, will have either 5 or 6 carbons. Mono means 1, saccharide means the base unit of a carbohydrate, so you're literally meaning one sugar. Some common examples: glucose, C6H12O6, is your body's chief source of energy. This is what gets broken down to be used as fuel for your cells. So this is a common monosaccharide. If you have two monosaccharides put together, that's called a disaccharide. Di means two. You actually have two units of sugar bound together. A good example of that is table sugar, which is sucrose. And then when you have many monosaccharides bonded together, that's called a polysaccharide. Poly meaning many. So many sugar units hooked together. And a good example of a polysaccharide is glycogen. The glycogen is a very long chain of glucose molecules that are joined together. And glycogen is a stored form of glucose. It's a stored source of energy that's kept in the liver until your body needs it or sometime in the future. Okay, here you have two monosaccharides being joined together, glucose and fructose. That's this one sugar unit. That's the second sugar unit. When they're joined together, you're forming a disaccharide because there's two units. Mono combined together, forming one sucrose compound, which is a disaccharide, and also water. Now we'll move on to lipids. These will consist of both carbon and hydrogen, and very little oxygen. And because they have a very little oxygen or other ions within them, lipids are considered to be hydrophobic. One of the main functions of lipids is it's used to store energy, also for protection and communication between cells. And there are many types of lipids. Fats and oils are very common. These consist of 3 fatty acid chains and a glycerol head that kind of joins them all together. These are the triglycerides. That's why there's 3 fatty acid tails. Energy storage molecules can be broken down whenever the body needs them. So these are a good source of storing energy. Waxes are lipids. They'll have a fatty acid chain and also an alcohol molecule. And these are some of the most hydrophobic substances that are known, used mainly for protection and such as waterproofing also. Now, a phospholipid molecule will have 2 fatty acid look like tails, and also a phosphate, PO4 with a negative charge, and the phosphate head. The tails are hydrophobic and the head is hydrophilic. Now, this structure is important because this something will form the cell membranes of cells. You have the hydrophilic heads pointing outside of the cell and inside of the cell, where you find water, and hydrophobic tails are going to be facing each other. So for a typical cell and cell membrane, you have a a phospholipid bilayer. You have two layers of these that wrap around the entire cell. This is what will help cells to communicate with each other. This is what will help control what goes in and out of a cell. A steroid is another example of a lipid. This is where you have carbon atoms arranged in rings. These are also used as structural molecules and used for communications between cells. Examples of steroids are cholesterol, testosterone, and estrogen. Here are some examples. The first one, the triglyceride, the fat. You have the three fatty acid tails here, and the glycerol head. Down here you have the phospholipid. This is what you'll find in cell membranes. The two fatty acid tails here, the phosphate head, the polar head, which would be hydrophilic. Non-polar tails would be hydrophobic. And you have these end to end, then the tails facing one another. And here's a chemical drawing of the steroid cholesterol. Next, move on to proteins. These are going to be long chains of amino acids. And because amino acids have nitrogen in them, proteins are always going to be very recognizable by these nitrogen molecules. They usually are the backbone of a molecule. And these amino acids are bound to each other by a special link called a peptide bond. This is unique for protein molecules only. The overall structure of a protein is determined by the order of the amino acids within the DNA, within the molecule. And the function of an individual protein will be determined by its shape. And some proteins are very simple, some are very, very complex. But all proteins, how they're shaped and what their functions are, will be determined by the order of individual amino acids found in the DNA. Now, these are the most versatile of all the biological molecules. It'd be really difficult to list every function that a protein has, but here are a list of just some of them. Help to speed up biological reactions, help store energy, help to move the body, help protect against infection, allow cells to communicate. And again, this is a very, very short list of everything that proteins are able to do. Here's some common examples of amino acids. This is a generic representation of an amino acid. Glycine, which is the simplest, aspartic acid, lysine, cysteine, so on. And there are only 20 amino acids that are found within the body, most of which your body will make on its own, but some your body needs to get through your diet. Last one of the four we'll talk about, nucleic acids. There are only two nucleic acids found in nature, DNA and RNA. That's actually what the NA stands for in these abbreviations. Ribonucleic acid, deoxyribonucleic acid. So those are the only two that are known as examples of nucleic acid. And of course, these two are involved with controlling the activities of cells because they contain your genetic code, your DNA. What makes you is what's found in DNA. A very standard image of a piece of DNA, that spiral staircase look, that helical look to them. This is an example of DNA because it's double-stranded. All right, now we'll talk about metabolism. This is all the chemical reactions going on within your body at any time. This will require various nutrients or fuel for your body to function, and it also will produce waste products. So metabolism is usually divided into two very opposite processes, anabolism and catabolism. Anabolism, this is the process by where you're taking simpler compounds and building them up into something more complex. This is used to manufacture materials for growth, reproduction, tissue repair, and so on. This is the building phase of metabolism. So taking two small things and building it up is anabolism. Many of these anabolic reactions are going to be dehydration synthesis reactions because you have water being removed and the other biological molecules are being bonded together. And the opposite of that would be catabolism. This is a process by which larger and more complex substances are being broken down into simpler. So you can think of this as a good way to keep these separate in your head is C for catabolism, C for catastrophe. When you have a catastrophe, you have things being broken down and broken apart. So same thing with catabolism, taking something very complex and tearing it down and breaking it down into individual smaller components. And many of these catabolic reactions are going to be hydrolysis reactions because you have water being added to something to help break something up. Hydro meaning water, lysis or to lyse means to break up something or break apart something. You're using the power of water to break apart a larger complex structure into smaller bits. That's why that catabolic reaction is usually involving hydrolysis. And here are some examples of both of those. This amino acid plus that amino acid plus the removal of water, dehydration of water, will form a dipeptide. 1 unit plus another 1 unit will give you a 2-unit product. So you are taking two simple things and making one more complex, larger thing. That is an anabolic reaction. The opposite taking something this complex, like 2 peptide unit structure, using hydrolysis to break it apart, and you end up with 2 smaller products. That would be a catabolic reaction. So anabolism going that way, and catabolism going that way. Now we'll move on to enzymes. In order for anything to get done within your cells, some compounds have to be broken down to be used. Some compounds have to be put together to be used. This is how all the individual organelles in your cell work. For both of those situations, taking something and breaking it down, or taking smaller things and building it back up, all those require chemical reactions. The problem with these kinds of reactions is most of these metabolic reactions will tend to happen pretty slowly. The hydrolysis reactions we talked about and the dehydration synthesis reactions can't happen without a little bit of help. Big functions of an enzyme is to speed along chemical reactions. The faster these reactions happen, the sooner the products can get made, and the sooner things can get done. So without enzymes, things will still happen, but much more slowly. So enzymes will make things happen a lot faster. And like we just mentioned, enzymes will speed up the rate of chemical reactions, and these are very specialized protein molecules that have special binding sites on them. Now, other biological molecules will bind to these enzymes and that are carried through the reaction. And once that reaction gets finished, the enzyme goes back to get more molecules. These are called substrates. So whatever binds to an enzyme is called a substrate. All right, because enzymes are going to be a binding system, you have enzyme binding to a substrate target. These are going to be very specific. Only certain molecules can be carried by certain enzymes. Substrates will compete for binding sites on enzymes. If those binding sites are all full, the enzymes are said to be saturated, because every possible binding site is being used. So that's why it's considered to be saturated. So molecules must wait for a spot to open up before it can go through its reaction. Enzymes can also be blocked, or what's called to be inhibited, which will prevent the substrate from binding to it. If they can't bind, then the reaction won't happen. And one good thing about enzymes are unchanged by their role in the reaction. And there's a good illustration of what happens. This yellow thing looks almost like a piece of macaroni pasta. This would be the enzyme, such as sucrase. This space here would be the active site. That'd be for step number 1. Step number 2, the substrate would bind to the enzyme. For this example, sucrose. The sucrose binding to the enzyme sucrase. You have hydrolysis being involved here, at least in this example. This will cause that substrate to be changed and being broken up. And the products you get are the 2 individual smaller components. So this is a good example of how you can tell what a compound is by the end of its name. If something ends with the letters OSE, that's going to mean it's a sugar. So lactose, sucrose, fructose, galactose. All those are examples of sugars, which are all examples of carbohydrates. If a term ends in ACE, ASE, that means it's an enzyme, and it targets that particular sugar. So sucrase will function with sucrose. Lactase breaks up lactose, and so on. So just by knowing little things like that will help you distinguish between what a compound is and what it does. Going back to the active site, if this were its only active site and it were full, it couldn't do anything else. Another substrate couldn't bind to it. This would have to wait until that one is freed. Alright, next move on to cell energy, and a very important compound, ATP. Good question is, how does energy get from the food that you eat, and has it turn that food into energy that your cells can actually use? This involves a process of digestion, taking food and breaking it down, and the energy gets released from that food. Now, cells can't use this energy directly. It has to be converted into a form of sugar, glucose. And then glucose is what is the main source of fuel for your cell. If cells don't get this source of fuel, then the cells can't function. If cells can't function, they will start to die. If cells die, tissues die. If tissues die, organs die. If organs die, you die. So glucose is critically important to your body's physiology. Now, glucose gets used by your cells through a very long series of reactions called cellular respiration. Now, during this process, glucose is combined with oxygen and is transformed within the mitochondria of your cells into a very high energy molecule called adenosine triphosphate, and usually abbreviated by the letters ATP. It is this compound that supplies your cells and then you with energy to function. So if there If glucose isn't there, then ATP can't be made efficiently. If mitochondria aren't working properly, then ATP can't be made properly. So without this compound here, most of the metabolic reactions in your body can't happen. Now ATP is made up of a base, a sugar, and a 3-phosphate group. That's why it's called a triphosphate. Now the phosphate groups are held together by very high energy bonds, and when those bonds are broken, you get a very large release of energy. That's how cells get their energy from this compound, by breaking those bonds. Now when that bond gets used, ATP becomes now adenosine diphosphate because you have 2 phosphate groups. So with 3, and then breaking a bond, you only have 2 left. So that's why it's ADP. Now that compound, ADP, is free to go travel and pick up another phosphate to become ATP once again. Here are some illustrations of the base, in this case adenine, the sugar, ribose, then the phosphate group. You have 1, 2, 3. That's why it's a adenosine triphosphate. If this bond here were to be broken, and that part gets broken away, then you have only 1, 2 phosphates, or ADP. Alright, that brings us to the end of chapter number 3 on biochemistry. Again, this is something that will be talked about in more detail in future chapters. So having a good understanding of how biochemistry works will give you a very good foundation for future lessons and future chapters.